On behalf of my co-authors, we proudly present this review of the clinical and translational research addressing the mechanisms, phenotypes, and progression of inflammation in eosinophilic esophagitis. Eosinophilic esophagitis is pathologically defined by accumulation of eosinophils in the esophageal epithelium, as well as epithelial damage marked by basal zone hyperplasia and impaired stratification. Examination of biopsy tissue has revealed a key role for type 2 inflammatory cytokines, most notably IL-13. In vitro work with human esophageal epithelial cells as well as mouse studies have shown that IL-13 acts directly on the epithelium to drive basal cell proliferation, impaired differentiation, increased barrier permeability, and release of eotaxin that drives recruitment of eosinophils. Given the central role of IL-13 driving the pathology of EoE, significant research efforts have been dedicated to determining the source of IL-13 in tissue and what triggers its release. Eosinophils can secrete IL-13, but there's more robust evidence from single-cell transcriptomic data of human tissue as well as animal models for a robust role of Th2 cells, mast cells, and type 2 innate lymphoid cells that are constitutively present in the esophageal epithelium and therefore capable of initiating disease. These resident immune cells are robustly activated by TSLP and interleukin-33, which are released by damaged epithelial cells. Transcripts and protein levels of both these factors are significantly elevated in EoE patient biopsies, suggesting that patients may be primed to respond to environmental challenges to the epithelium. These environmental challenges have also been extensively studied, primarily in mouse models that facilitate precise dissection of environmental and dietary exposures. Several dietary components have been identified as allergenic in EoE, which is supported clinically by patient improvements when implementing elimination diets. Moreover, experimental evidence suggests that detergents, which are present in dishwashing soap and toothpaste, are capable of disrupting the epithelial barrier and directly triggering IL-33 release. Prolonged inflammation is thought to drive the onset of fibrostenotic tissue remodeling that's observed in EoE. A broad and deep body of research has identified clinical phenotypes and future therapeutic targets, and these exciting findings about the relationship between inflammation and fibrosis are paving the way for better clinical evaluations and innovative clinical trials. Overall, advances in understanding and dissecting the mechanisms that drive inflammation in EoE and the relationship between inflammation and remodeling has led to modifications in the diagnostic criteria and the development of effective therapeutic strategies for EoE. We hope that you find our manuscript informative and thought-provoking.